Okay, chapter 10, viral genetics. This is a short chapter, so you guys ought to have fun with this. It's interesting information because one of the things about viruses is that they are non-living things. They're pieces of protein and genetic material. People often refer to them as particles. And of course, they're medically important because viruses are responsible for many diseases. When we have an infection, the infection is usually initiated virally, but then bacteria come in and take over the environment that the virus has disturbed. And so it's really important to understand how viruses replicate and how viruses do the things they need to do to make new viruses. One of the things you need to know is that viruses are not alive. They don't reproduce. They trick a host cell into making more of their little armies. So viruses, one of the things about them is that they share nucleic acid or DNA, RNA genomes with us. And so that suggests that evolutionarily we co-evolve. Now that's a whole separate class that's really interesting, but hopefully in your future you'll be able to learn more about evolution of viruses. There are viruses that infect specifically only prokaryotic cells, and there are viruses that infect eukaryotic cells, and they rely on that living cell to replicate. A virus cannot infect and copy itself in a dead cell. So just some details. The size of the genome or how many genes each virus has is variable. The genome can be circular or linear, which is kind of weird. Circular suggests that those virus particles are probably infecting what? Bacteria or prokaryotic cells. The type of nucleic acid, of course, can be double-stranded DNA, which is what we're used to, but sometimes they use single-stranded DNA. Crazy. Double-stranded RNA, which most people are like, double-stranded RNA? Well, it does exist, and viruses carry them. Or single-stranded, this should say RNA here. I don't know why it says DNA. That should say RNA. Sorry about that. Here's some examples of different viruses. The first set of viruses up here, all the phage viruses, these infect bacterial cells like E. coli. There are viruses that infect plants, so like tobacco mosaic virus infects plants. There are actually viruses that infect insects, mammals that we're used to. There are viruses specific to fish, you name it, there's a virus for it. And then, of course, there are the human viruses, things like Epstein-Barr, adenovirus, herpes simplex virus, and there are a few versions of that, human papillomavirus, and HIV. So all viruses share a few things in common. First of all, they have an outer capsid, okay? An outer, not exactly membrane, but protein shell. And this shell is called the capsid, okay? And the proteins of the capsid are called capsomeres. And so capsomeres are very specific. It's a specific group or protein type that you'll find encasing the genetic information of viruses. Many viruses have that capsid as their envelope, and the outside may or may not be studded with something that we call glycoproteins. And so in the last lecture, I kept drawing this little robot-looking like thing. Sort of robot-looking. It looks terrible. Sorry. The outside of this phage is the capsid. All right? Now, the genetic material inside of this phage might be RNA, single-stranded RNA, or it can be single-stranded DNA for all we know. So this particular not organism, but virus is put together with protein capsid on the outside and nucleic acid inside. On the outside of the capsid, there might be lots of what we call glycoproteins. Now, in a phage, you don't necessarily find these, but in a human viruses actually look a lot like phage. They just don't have the little foot or the little tail fibers. So these protein spikes or glycoprotein fibers are going to be something that you find on the outside of the capsid. Now, in terms of looking at the structure and identifying elements of the actual virus, there are a number of things that you might look at. what type of proteins here. We identified glycoproteins, but there can be other types of protein there as well. We don't need to name them right now. So here's some examples of what these proteins look like. This is a tobacco mosaic virus. It has a helical capsid. It looks kind of like a churro, quite frankly. And then here's the nucleic acid, RNA in the middle. So that would be like the cream inside the churro, except for, you know, it's not going to be delicious because it's obviously a virus infecting a tobacco leaf. Pardon me. Adenovirus is a human virus, and as you can see here, it has this polyhedral shape, kind of like the head of a bacteriophage. And inside you have the nucleic acid, DNA in this particular case, double-stranded DNA. And then each one of these proteins is a capsomer. This is a protein fiber with a knob on the end, and this is very characteristic of adenoviruses. Now, the influenza virus is even more complicated. It has this kind of polyhedral capsomer-based envelope outside of an RNA genome, but then outside of that, you have a viral envelope, something that is spiked with tons of glycoproteins. So this is a very complicated virus. The influenza virus has a lot of protection and a lot of ways of hiding from the immune system based on these particular glycoproteins and this viral envelope on the outside. So this is a much more complex virus. Now, viruses reproduce usually in 6 steps. The first one is when the virus attaches itself to the host cell. Basically, what the virus is doing, let me go back one slide, is sometimes these spikes on the outside are used to identify and attach to the host cell. So let's say, for example, in influenza, these usually infect lung. It can infect the throat. It infects mucous membranes. Oh, you know what? I just figured out if I push harder, it doesn't skip. Oh, learn something new every day, right? And so these little glycoprotein spikes assist in identifying the tissues that the cell needs to infect the right cell type. The next thing that happens is the virus has to enter the cell, and there are a few mechanisms for that. The virus itself doesn't necessarily always have to enter the cell. The virus can just inject its DNA into the cell like bacteriophage do. Now, after that, you have integration of the genome, if that's a part of its particular infection mode. If the virus is going to be reproduced, new viral components need to be made. New viral protein coats need to be made. New capsids are made. And then you have the assembly of what we call infectious particles, the assembly of the new virus. So you have to have the protein coat, maybe that viral envelope produced, new DNA or RNA put in the middle, and then release of the virus. That's when you start sneezing or coughing, or the transmission route is kicked off. Now, the reproductive cycle in attachment, there are only a few specific cells that every virus actually attacks. It just happens that they're really good at finding those cells. So, like I said, proteins on the virus help the virus identify and interact with the cell surface. The reproductive cycle starts with entry. The genome enters the cell. Sometimes the whole bacterial envelope enters the cell. But in this particular example, we're just going to think about the viral genome entering the cell, okay? Usually the viral genome in bacteriophage is the only thing that enters. It enters alone. And then the viral genome with associated proteins. This is more phage. And this is more mammal, and so forth. Some viral proteins are made immediately. They're synthesized immediately. And that's where you start getting new things happening in the cell. That can kick off integration of the viral genome into the host genome. Not all viruses do this. Varicella virus does this, so the virus that gives you chickenpox does this. Human papilloma virus does this. Human papilloma virus can integrate in tissues and lay there dormant for years. and then later in life emerge, resulting actually in cancer. This is one of the ways cervical cancer occurs. And then you have things like herpes virus, just simple cold sores. When you're stressed, the viral genome becomes active again and you end up developing sores. Integrase is the enzyme that allows the new genome to insert itself, let's go back one slide, into the host. So if you have a cell, remember, if you're dealing with a mammalian cell, the virus has to not only get into the cell, okay, so here's the viral DNA. It's going to have to do something to interact with and destroy the DNA of the host, all right? And when that happens, that's a mess. It's killing, bad Dr. Val, look at this. Here I am making circular chromosomes in the human nucleus. Ugh, bad job. Okay, so the chromosomes will be destroyed, but integrase, if this virus is going to integrate into the human genome, the virus DNA, the viral DNA, gets into the nucleus, and integrase, an enzyme that is in the viral DNA, is expressed, and it allows an individual chromosome to accept or integrate the viral DNA into its chromosome. So, we're not going to go into a whole lot of detail yet about integration. Let's talk a little bit about bacteriophage. Now, bacteriophage was the first really well understood type of viral infection. We know that bacteriophage, is a temperate virus. What that means is that it'll do one of two things. I think of it as it's like a fussy virus. It has one phase that's called the lytic phase, and this phase causes the host cell to explode or lyse. So lytic equals lysis. Then you have the lysogenic phase. Do not confuse lysogenic with lysis. Lysogenic is where you have integration of the host chromosome and the viral chromosome to make what we call a prophage, okay? This does not lyse the cell, all right? I cannot say enough how this does not lyse the cell, all right? You must understand that this does not lyse the cell. Lysogenic simply allows the bacteriophage to integrate its genome into the infected cell. And some bacteriophage don't even have a lysogenic cycle, all right? And these are known as virulent viruses. If there's no lysogenic cycle, where the virus can hide its DNA and hide out until another time, what it's going to do is just kill cells in the organism. And as the cells die, that's called virulence. It's damaging or hurting the organism. So here's a really nice figure. Actually, this is one of my favorite viral figures. It's simple and it's to the point. It's identifying the difference between the lysogenic and the lytic cycle. Okay? So first of all, in this example, let's start with the lytic cycle. In this case, the bacteriophage infects the cell. It injects its DNA. The DNA enters the cell. Genes are expressed. As a consequence, the bacterial cell, its DNA gets all chopped up, and the phage DNA directs the construction of new virus particles. So basically what's going on here is the phage's DNA is taking over. It's killing the host DNA, and it's using all the enzymes in the cell to do the job of making new viral particles. Then it finishes the job by causing the cell to lyse, to explode, releasing all these particles into the environment. So if you think about it, if you have a series of cells sitting next to each other in a petri dish, I guess I could have made the petri dish round, and each one of these cells is infected by a phage, as these phage lyse and the particles get released into the environment, adjacent cells can become infected and they're going to lyse. And you basically have a region of death growing across the petri dish as the bacterial cells as a consequence of the lytic cycle. So I'll start the new video where we do the lysogenic cycle right now. See you in a couple minutes.